You go from a single cell layer to multiple cell layers to start specifying the pattern of the embryo, so which is where the head is, where the tail end is going to be, etc. You start specifying organs, so the heart, for example, and the central nervous system. So this is such a critical period of development, it seems, and we know almost nothing about it in humans. So this is also a critical period where if anything goes wrong, you're going to get miscarriage or congenital defects in the children born. It's such an important part of embryonic development to study, about which we know almost nothing. So there has been over the last few years, increasing feeling that the 14-day limit is too restrictive, where in fact, it may even be ethically unjustified to have that 14-day limit because we're missing so much valuable information that could help couples have healthy children.
We need to understand what happens in early human development, because unless we understand that, we can't think about treatments that do generate human genetic disease, aneuploidy, point mutations, developmental defects. And at the moment, for example, there is a limit on embryos, culturing human embryos for 14 days. Do we need to extend that? New technologies are coming along where you can put human stem cells into mice, for example, and study their development. Now, that research will have an impact in the future on our understanding of those diseases. So they are, in my view, important, because if we understand those diseases, we can think about treating them.
These are the organoids.
Science is happening so quickly, it's quite difficult to have the right ethical debate before we're faced with the decision, should we use this technology or not? So broadening that category and having places where the science is possible and you may need to come to a decision, and leaving it open so that you can have dialogue about what happens next is going to be incredibly welcome, I think, by everyone, scientists and the public alike.
6 months? Yeah. Wow. So something is wrong with this cell. It's probably gonna die.
Then you can also zoom time.
We also discuss in the guidelines another new development really over the last 5 years, which is these stem cell-based embryo models. So these are not embryos, they are models. We hope that they will be valid models, and that's one issue. They need to be validated against normal human embryos. But if they are, then you could, rather than using human embryos in culture for more than 14 days, you could use these stem cell-based embryo models. And these are, you can make many of them. You can do proper controlled experiments with those. So they would be, any committee should say, well, if you can do your experiment using a model, then use the model.
We've been talking more and more about the 14-day rule, for about 4 or 5 years now. We've done public engagement at the Science Museum asking what people would find acceptable in relation to human genome editing. And when you talk to people about these things, when they see the scientists on the TV, it sort of takes away a lot of the mystery and the fear.
Scientists tend to focus on these narrow questions. I want to answer this specific problem. Members of the public, when I've been involved in dialogue exercises with them, they tend to have a much bigger picture. They're concerned about other issues or broader issues. And so I think it's very valuable to have this conversation so that scientists get to appreciate really what concerns the public get to understand what's doable and why scientists really want to focus on getting this done.